is basically the establishment of in vitro cultures of new nematode isolates, which are from Italy, the POP16, and from the Azores, EZ29. After we establish these cultures, we do the production and the formulation for field trials, and we also test the quality of these nematodes for the field trials with our standard methods. As a special gimmick, we also have Bacillus thuringiensis galleria, which is known to infect Popillia japonica, and we offer that for trials. To produce nematodes in vivo, we need insects, mainly Galleria melonella. Here an insect is drawn, and as you can see, they are surrounded with foreign bacteria and also have a lot of bacteria in the intestine. These bacteria, they inevitably enter the hemocell when the nematodes enter through the anus, the mouth, or through the spiracles. The nematodes also bring in their own symbiotic bacteria, here in red, and these bacteria are spit out. And the bacteria which are coming in through the wounds, they are eliminated by the insect's immune response. So what we've got inside an insect is a sterile environment for the production of bacteria as food for the nematodes. And the nematodes basically feed on bacteria, not so much on the insect tissue. That's why we can also produce nematodes in vitro. We just have to provide a sterile environment with nematodes and the bacteria. To do so, we have to isolate the bacteria from the nematodes, and we also have to get sterile nematodes. And we infect insect larvae and then take out a drop of hemolymph to get the symbiotic bacteria from the infected larvae. That's quite an easy, straightforward step, simple standard microbiology. To get sterile nematodes is a bit more difficult. There we work from gravid females, which contain a lot of fertilized eggs with small nematodes in there. And these eggs are surface sterilized with bleach, and then the worms hatch in a sterile nutrient solution. And if bacteria have actually entered the solution, that we get turbid, so we wouldn't take nematodes from those turbid vials, but from the other vials we take the sterile nematodes, combine them with the bacteria on agar plates, and there we have our monoxenic culture. And this monoxenic culture on agar plates can then be transferred after the nematodes have multiplied into small shaken flasks, and from these shaken flasks we can work to even larger volumes from liter to cubic meter scale. For the liquid culture, we always inoculate first the bacteria in a vessel, they propagate, then we add the nematodes. The nematodes inside the tank will open their mouth, start feeding, and then multiply. And by the end, we've got about 200,000 nematodes per milliliter, and we started the process with 2,000 nematodes per milliliter. Yeah, so this is the task which we do for the project to provide sufficient numbers of nematodes for field trials against the Popillia japonica larvae. Thank you.